Welcome to our Microbiology Journal Club. This is our online journal club where we talk about microbiology papers, as a journal club is, as promised. Today we're going to talk about two papers having to do with taxonomy, one that's a little broader in terms of it moves beyond microbiology to think about taxonomy as a field and as a topic of study. And then we're also going to talk about a paper that specifically discusses some of the challenges of bacterial taxonomy, which are many, which we'll get into. And today, actually, we're going to be led by Nicole. So at this point, I'm going to stop talking, and I'm going to turn it over to Nicole.
Hello, everyone. So I am Nicole Sictio, and you've probably seen me on a couple of the micro JC meetings before, which, so I'll just describe. I'm very much firmly outside of organismal biology, which is what a lot of taxonomy centers on. I've spent a lot of time in the last few years looking at 16S and ITS2 amplicon sequencing data. So a lot of these, I share a lot of deep guilt about being flippant over taxonomy. That is inappropriate, and also recognize, you know, what legacy you leave behind in terms of ambiguity and having to revise how we use language to point to organisms for what Dave describes in one of his papers as manuscript continuity, how we actually point to things for the public at large and stakeholders who will now have to interact with all the biodiversity, microbial and otherwise, that we have either through things like climate change, anthropogenic modification of land, and the context of disease, which is where a lot of us learn bacteria names and has sort of historically set the context for classification schemes that have not worked out quite so great. So I think since it's a small group, Karen is here. I'm just gonna mute so Karen can say hi for a bit.
Oh, hi everyone. My name is Karen, and I'm a postdoc at Trinity Western University in Vancouver. And I study, well, at the current moment, I study the effect on Pseudomonas on C. elegans, certain C. elegans genes. And I share Nicole's I share Nicole's thoughts about like being flippant about taxonomy and so in that sense, I can talk a little bit more about it later, but I definitely feel the same way. And so it was important for me to read, and I have some ideas and some thinking that I'll also share later, and I'll turn it over to Laura.
So I'm Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island. I really enjoyed reading both of these because in my teaching, I usually do the what we call Bio 104, so our spring semester of general biology, which is a bit more ecology and evolution focused. And I teach the lab section a couple of times, and we have a challenging task of trying to figure out how within a laboratory course setting to teach about biodiversity, to teach about the tree of life, to teach about taxonomy, and to get students to actually appreciate that taxonomy is not just this like, and Holly Bick mentions this in her paper, this kind of like dry, you got a dude in Victorian garb just sitting at a desk kind of painstakingly drawing things. And I think that's how it's perceived. So I think both these papers brought up some interesting points that I'm looking forward to talking about.
Okay, so back to me, and I think we should probably describe what the papers are before we get started. So I'm just going to give like the fast summary of both of them because there are two papers, and I think I want to have a very interwoven discussion, but because there are listeners here who may not have read both and probably want to know what they're in for, here's the quick play-by-play. So Holly Bick's paper, Let's Rise Up to Unite Taxonomy and Technology, is very much a perspective piece. Ooh, I can hide behind this. I won't do that. A perspective piece on very engagingly written to talk about societal and, as we have mentioned, even among scientists, the antiquated visual connotations of what taxonomy is, the current state of recruiting people into that branch of biology, which is this feeling that we are losing a lot of the specialist knowledge to actually do hardcore deep inspection of multicellular eukaryotes, mostly, that have morphological characteristics that can be used to create biological and taxonomic classification schemes. How this is still a relevant and needed practice for basic discovery of biological and biochemical processes embedded in diverse pieces of life, as well as creating sort of this global awareness of what we have on the planet and what we don't have with respect to biodiversity. And also addresses something that we get into with the second paper, which is the deluge of DNA sequence-based and genomic data. And how do we reconcile this with traditional methods of taxonomy? How do both fields exist adjacent to each other? And how do we manage all that data? Because both traditional and sequence-based taxonomy are data-dense bodies of knowledge. The second paper, divorcing strain classification from species names, takes us to some very interesting places like phenotypic classification hell and taxonomic limbo. And it is set up also as a perspective piece by one of our intermittent participants, Dr. David Baltrus, speaking from a very bacteria-centric side. So Dave gets into some of the procedural considerations for defining a type strain and how you would go about doing that, which was new to me because I have spent most of my life taking things out of bacteria that weren't meant to be expressed in them and looking at them, and then looking at a list of things on an Excel spreadsheet that are supposed to be in a certain compartment. So this idea of looking at something in that much detail outside of my field completely, but then gets into sort of a couple of example organism perspectives, including Helicobacter pylori, which pretty much strain-wise tends to stick around in the gut and has had sort of nice evolutionary and niche characteristics that allow things to be clustered very tightly, whereas the Pseudomonads live in all the places and do all the things, and that makes it very hard to do attribution-based classification of organisms. So Dave kind of gives us this walkthrough of how Pseudomonas syringae has led us down the road to phenotypic classification hell and taxonomic limbo, and then proposes a genome-based scheme that is cited in one of the papers that he includes in his references to sort of reimagine taxonomy independent of names and based on something a little more objective and quantitative, which is average nucleotide intensity, identity, and genome similarity. So that's sort of like, that was 5 minutes. Okay, that was not bad. So let's start with this. Now that we know what the content of both papers are, is kind of addressing the fact that, so Holly starts her paper with talking about taxonomy as this thing where you see somebody at a desk. And these days I've watched some of the entomologists here, so they're not in Victorian breeches, they're usually in Lululemon spandex with hard rock coming through the radio, looking at a dissecting microscope. But I mean, the posturing and the intensity of having to look at minutiae, which is usually genital plates and bodily structures, and try to come up with what is this thing, or maybe it's not mature enough, so I'm not going to get a good idea. Those are still very much the visual trappings of that type of science. But what I think is really interesting about both Holly's paper and Dave's paper is they basically provide a lot of material to sort of imagine, well, or enact a discussion about taxonomy in a place that I'm sure all of us are very nervous as practitioners of learning, or at least I will be next semester, because Karen, you're already teaching, and Laura, you have been teaching, and we are teaching in fields that are undergoing continuous knowledge revision and shifts and debates in how we handle data, how we talk about the biology we have, and we still teach. So I was just kind of wondering about, at this point, the group's perspective on how you viewed these papers overall as vehicles for student engagement in the topic of taxonomy, bacterial or otherwise classification, and also acknowledging in front of students, which is sometimes difficult, that there's a lot of these uncertainties about how we should handle things that we share that we don't have clear answers on, but at the same time, it's exciting to think about. So I'm going to open up the floor to that sort of perspective.
So I'll see if I can answer that. First of all, I saw Dave popped in, so hi, Dave. Okay, so I'm in the context of teaching GenBio, I think I mentioned that we have to, one of the things we try to convey is you're trying to get at these goals of having them understand biodiversity, and you're having them understand relationships among organisms and evolution. And it's really hard to do it because I think they just see taxonomy as this boring list of things, and you go through a key in a flow chart, and does it have that, does it have this, does it, okay, does it go there, does it go that? But one of the things that Holly pointed out broadly in her paper is that having this very specialized knowledge of the, if you're not talking about microbes now, if you're talking about, insects, for instance, having this very specialized knowledge about the morphological features that are typically used to classify or figure out who's related to who is a really important piece of information to intersect with the genomics, because all of that is the product of, this lineage. In some cases it's adaptive, in some cases it's not adaptive, and you can talk about all of those things. So you could say, well, why does it have this? Why does it have that? What's the circumstances that it's in where we've got these things going on? And I think that one of the things I thought as I was reading Holly's paper is, what can I pull out of this? How can I use this to help me figure out the inroads, the access points for students to actually engage with taxonomy in a way that's not just, here's a list of stuff, these are chordates, these are this, these are that, to actually get them to think about what would be the purpose of cataloging these differences, what do all these differences mean? Yeah, I'll stop there for the moment, I think. I have more thoughts, but I'll stop there.
Okay, I really like where you went with that in terms of thinking about how to draw students into it's more than a list. And I think when Holly hit on the biodiversity thing, this is a very interesting intersection of taxonomy with what we want the data product to be. I've noticed that Luke has joined us. So Luke actually works in a lab that's just down the hall from me. So I'm just going to say hello.
Hi.
Luke. Hey, Luke.
Can you hear me okay?
Luke is working on characterizing a bunch of well, actually, you can introduce yourself after I just briefly say something for 30 seconds, getting back to biodiversity. And that is that with biodiversity, there's a movement towards how do we figure out what to save? How do we figure out what to prioritize? How do we figure out what to characterize? So there's this triage procedure, as well as the whole, well, if the list of all these types of flies colloquially are there, is there a problem? Because they're still there for this particular anthropogenic land modification. But if we don't have a good scheme to actually look at the finer detail of what's actually there, and we don't have, people looking at what compartments of, say, a water column are we finding these, or under what seasons are this a problem, or when was the last time we saw this? The minutiae of figuring out what we're losing, what we prioritize, depends very much on taxonomy being as conclusive as it can from this morphological standpoint, from looking at these ecologically defined traits, which is something that Pseudomonas syringae doesn't really allow us to do, but there's other organisms that do allow us to do that. So I think the idea of thinking about conservation and the idea of thinking about this contrast, because we also have genome plasticity and adaptation that can happen at faster rates in the bacterial realm as well. So we still don't really Like, I don't really know how to talk about extinction of bacteria when I talk about soil, because we don't even know most of the time when they're awake and doing stuff and not, because they can do that thing too, where they nap until the substrate shows up. So these ideas of like, is this just a list? Or is there interactions embedded in this list? So, I mean, this is not the most concise way of talking about student engagement with taxonomy, but I think these ideas of talking about process that's embedded in the list is one of the ways to actually bring that back in. So I'm gonna stop with that, and I'm gonna let Luke introduce himself briefly.
Hi, can you hear me okay?
We can hear you.
Okay, good. I have kind of crappy internet where I'm at, so I might be cutting out. But anyway, I'm Luke. I'm a master's student at the University of Northern BC. I work in a lab beside Nicole, and I am trying to functionally characterize some oxidative stress proteins from the mountain pine beetle. And I'm really excited to listen to this discussion on these papers. I don't have a lot of hot takes, but I'm really excited to hear what all of you have to say.
Thanks for the intro, Luke. So we were just getting into a little bit of discussion on the two papers and ideas that have come out of it as a vehicle for student engagement and discussing taxonomy in the classroom setting. So Karen, I wouldn't mind hearing from you next, because I think we're kind of going in a circle or pentagon or something. So
It's hard to go in a pentagon when it's like in a row down at the bottom. But I totally agree in terms of student engagement. For me, is very much about describing the process. Like one of my favorite things to do when I teach is to present historical context and actually humanize the material that I'm teaching. So I usually place an emphasis on like, okay, I'll talk about the person who discovered this, or talk about the process that they had to go through to establish or to get to a protocol in terms of teaching a lab. If I can find the stories and a little bit of background, if I can place that into context, I think then they're able to connect with the material much more, rather than just considering it as, okay, it's just something that I have to memorize. Well, in terms of like, that's the way that I would approach it normally. But in terms of teaching taxonomy, I'm unfortunately don't have a lot of experience in teaching taxonomy, other than, okay, like what Laura was describing, where you have the different, okay, this bacteria has this and does not have that, and therefore goes here, and there, and I don't know if this is going to come into left field, but as I was reading Holly's paper, I was thinking that maybe it's an evolution of the way that we classify things. Like we used to do it morphologically, and we used to do it based on these biochemical tests. But what if so I'm wondering if in the future we're heading towards this direction where we're classifying it genomically instead. And like, as I was thinking about it, I was like, well, but if we do it genomically, then we lose that phenotypic, like you lose whether or not there are phenotypic differences in terms of the way that they are expressed. So because the organisms can be completely identical, but you have variation in phenotypic expression, like by doing it genetically, you would lose that ability to classify those phenotypic traits. So it kind of seems like for me, it feels like there needs to be this marriage between phenotype and genomic traits. But then, like Holly was saying, that in order for that to happen, you need to have a collaboration between both groups instead of just having it one way or the other.
So
yes.
I'll step in here.
Okay.
So I'm somewhat agnostic about the whole taxonomy issue. So I think phenotypes are always going to happen. We're always going to measure them. People are going to be interested in them. And then it's just a question of what framework we hang those phenotypes on. So whether we use it to define what things are, or whether we define things and then explore phenotypes after the fact, I think is the relevant question for this. And I point to like, that paper was written mostly out of frustration for a variety of different ways. And I try to come at it from a more practical viewpoint of, sure, like we have this system that's been set up for a while, and it's worked really well in some cases, but it's failing in a lot of other cases. And especially in things like microbes and when talking about phenotypes. So it's great if you have, E. coli, and you know that E. coli has certain phenotypes that have been established. But when you actually, when you look at other species that aren't that well fleshed out, there may be a species name, but that doesn't mean that that trait that we've defined as an important trait is actually really important for the species. It might be different, and it might be physiologically different. It might be phenotypically plastic. It might change. And so when you set up what I came around to in the end with that paper was that if we set up this framework that can't grow and be added to, but where we have to completely overhaul everything every time something new gets discovered, I think it's a failure on our part because we're not anticipating what's going to happen. And so phenotypes are great, but that doesn't mean, you find one exception and then you have to change everything again. You have to rewrite tons of stuff, and you lose a lot of knowledge to history, because when names change, you lose linkages sometimes, and it's difficult. So I don't think phenotypes will always be interested in defining what things look like and where they live, and getting the metadata to actually figure out, how the interesting ecological questions. But I think what I was trying to get at is we need a more flexible framework to put that in. Because right now, like even to define, this is only bacteria, but to define a new bacterial species, you literally have to do DNA hybridization, which is kind of insane if you have the genomes. I have to do this now for one of them, and you literally have to do the hybridization between genomes, even though like the data we want from that you can get other ways. So it's yeah. I think that the challenge with students and bringing this back to the classroom too is that you teach students things like, I'll teach the original transformation experiments with Avery, McLeod, and McCarty, and Griffiths, and they were working with pneumococcus, which is now not called pneumococcus anymore. It's called streptococcus. And so when you teach students, do you teach them pneumococcus because that's what the textbooks say, or do you teach it streptococcus because that's what it is now? And so it's I think if there's a way to minimize the amount of for non-experts and students that are learning, to minimize having to go that's an important point to consider too. But
okay, Dave, I wanted to ask you, we lost you a little bit towards the end there, which might just be related to
no, somebody's trying to call me from Vegas, so
oh okay, maybe that's why. I wanted to ask you too, because in reading your paper, I just wondered if you would, so you're making a distinction between classification and nomenclature. Yeah. So I had to make sure I said that right. I was like, that didn't sound right for some reason. Can you talk about that? Because I think I have an idea of what you meant, but what do you mean by these are two different parts of taxonomy? You've got classification. Yeah, I think you're kind of getting at that with the idea, and I think Karen might have been headed that way slightly too. But I just thought, could you talk a little bit about the difference between those two?
Yeah, so I was trying to, when I was sitting down to write that, I was trying to think of what value do names have, and I came around to kind of what was being talked about before, where if you have a name and that name is basically shorthand for phenotype. So if you have, we call something E. coli, you kind of know what E. coli is by calling it E. coli. So you know kind of how it's going to grow, its temperature range, and those are all characteristics that have been defined in order to call it E. coli. But we can just now call it E. coli and related information. You find something else that's called E. coli, you pretty much have a good idea for why that's valuable. So the nomenclature part really is like, if you're in a hospital and you've got C. diff, it's good to have that, call it C. diff rather than something else, which we might not be able to easily, it might be more confusing. The names don't necessarily, so when I started thinking about that, I'm like, all right, names are good because they give us shorthand for other characteristics, but that doesn't mean that the names need to say anything about classification and how organisms relate to each other, right? Because E. coli is different than Salmonella, but they're pretty closely related, all things considered, right? Even though the names, the shorthand, we can call them Enterobacter or whatever they're called, Enterobacters, but they're called something different, right? So the nomenclature gives a shorthand for phenotypes, but it doesn't necessarily say things about if you find a new isolate, that doesn't tell you anything about how it's related to anything else unless you know the whole framework for understanding it. At least with genomes, there's an inherent way, and we've developed tools to be able to take a new genome without even having to call it anything and place that in a relational way against other genomes that are out there. So we may not know what the name is, but we can place that within the framework to understand expectations for what we might expect based on the phylogeny or based on what else it's related to. So we may not know what, you may have a new isolate, but it may be closer related to E. coli. It's, might not be called E. coli, but the fact that it's placed in the genome next to E. coli kind of gives you an expectation for how it might behave. And so the name is separate because you don't need to have a name to have an expectation for how its cousins behave and therefore how it might behave. And you don't need a name to know who are cousins based on genomes, basically. So hopefully that makes more sense. But I just got at it from, I have a lot of isolates that are called Pseudomonas, and they're not technically called Pseudomonas syringae because they're not from plants. And they're from the environment, and they're kind of phylogenetically considered. And so I would like to be able to place them in a genome and say they should behave like these isolates, but formally I'm not comfortable calling them syringae, Pseudomonas syringae, because they might not have the phenotypes that Pseudomonas syringae is known for, or as you guys probably, if you read the paper, I'm writing something now, or this term's probably going to get cut out, but I'm calling it the Rube Goldbergian morass of Pseudomonas syringae, because it's just, we keep adding on different things to define them, and then it becomes this monster of a thing in the end where you lose track of how to actually define anything. So at least if you have genome information, you can classify it without having to name it, but you have to kind of classify it to be able to name it too. So there's two levels there that that go together, but they're not inherently linked.
Yeah, I think
hopefully
that helps. Yeah, that's kind of what I had gotten from it in terms of what you're saying. And I mean, I think from a bacterial perspective, I think that's very helpful. Yeah, the Pseudomonas situation looks like, yeah, earlier Nicole was referring to it as oh, I can't remember. It's either, it was great. One was hell and one was limbo. And I was just envisioning these 7 Dante circles of taxonomy. Which one are you in? But I think the thing that's useful too about the continuity of within a research setting. So there's 2 points that I think both Holly's paper and Dave and your paper make, which is because of our confusion about bacterial taxonomy on the one hand, and because of our lack of interaction with old school taxonomy in non-microbial systems, we are missing out on a tremendous amount of information that is work that has been done, work that has been published, but work that is currently not accessible because of either, in the case of what Holly mentions, it's not been digitized well, or it's not been organized well. And so if you're studying this particular insect, there could be a whole wealth of information that somebody else did 100 years ago, but you can't find it because we have not figured out how to incorporate that information into the things that we're used to searching, database resources and repositories of things. And on the bacterial side of things, I know from what the paper that we just submitted, it's Halobacteriovorax marinus, which started out as Dellavibrio, which then became Bacteriovorax, which then got renamed Halobacteriovorax. So I've got a part in the paper where I'm okay, don't freak out, but it was this, then it was this, then it was that. So if you're reading about this, it's probably not that, it's probably this, but it's not that. Got it? And so I think that both papers kind of get at this idea of simplifying access to information and making sure information is consistently available as you're moving things around. And I'll stop there and turn it back over to Nicole. I kind of took over. Sorry, Nicole.
That's fantastic, actually. I'm really glad you did that because there are things dropping out of my brain right now. And I'm really, really glad that you brought up the section that Holly mentioned about qualitative predictions of organisms in terms of like that metric of classifying, you know, non-bacterial things where you have, you know, these key sort of features. And I've seen with things like the radiolarians, I mean, even at the microbial level, you can have these morphological differences. And I just wanted to take this opportunity actually to shout out two things from social media that I think are sort of starting to help bridge that. I don't think they're necessarily real, like, hardcore visual repositories, but there's the Biodiversity Historical Library, which you may have seen tweeting as BHL, which has scans of a lot of natural history things. So there are people who are trying to get this stuff preserved, getting documents in electronic format. The other one that's sort of amusing is a bot called Old Fruit Pictures, which is actually the USDA's archive of like agricultural, and some of these are no longer really marketable, like fruit that were parts of USDA experiments or categories of, so keep these in mind. Like there is this type of visual record of some of this data, and the fact that it's getting up there means that there is something in the cloud up there that we can actually link to data management. And I think that's actually something I want to come back to later, because that's the different access of interdisciplinarity in the sciences that some of us, I feel very icky talking about it because my hard drive looks awful. And it's a best practice thing that we actually, like, it needs to be a thing that we are giving early career scientists like a capacity to deal with when we deal with all this stuff too. But I also just wanted to come back and sort of bundle a lot of the things that Dave and Laura have said in the last few minutes about when names are useful. And Dave's clearly in the paper, and as well just now in this conversation, highlighted that there's different sectors, different stakeholders, and different policy interpreters. Society is a policy interpreter. So having a name that you can hang on a disease for the purpose of conversational continuity, to describe something, to let people know why this has an impact on our environment or our social structures, we need names in that respect. We need them in biodiversity contexts because numbers of names do matter in some indices, you know, not just the ecological context, also though that comes in. But Dave is also leaning towards the part where I think we should go and talk about phenotypic classification hell and taxonomic limbo of Pseudomonas in the next phase of this discussion, which is that the genomes provide this objective quantitative bridge. We're actually looking at an attribute of an organism that we can say differs by a certain fraction and allows us to point to them based on this thing that's not really going to change because we have genomes, when you can get genomes. So I saw a tweet last night for somebody looking for classification schemes for resupinate and crust fungi, which is not trivial. And those are also things that you can't really get genomes for because we have tons of piloderma in the site that I'm in, where you dig in a couple centimeters and you see this stuff that looks like crumbly, bright yellow rice noodle. It's a fungus. It's a mat former. It doesn't really produce fruiting bodies, though. So my chances of being able to sequence a genome with that, not great. I know the JGI is doing the 1,000 Fungal Genomes project. I suspect that it's going to be underrepresented for key symbiotic mycorrhizal taxa for years to come, because you kind of need to culture that with a partner in a lot of cases. I might be getting that wrong. I know that's not always the case. We can get Lacarian culture. This is a bit of a departure, but I thought I would bring it in because the discussion is about to go very Pseudomonas-y. But I wanted to kind of bring light to the idea that genomes are still a bottleneck for some other types of organismal biology.
But I think I think it's instructive to look towards you know, the Banfield lab stuff, where we do have a ton of genomes that are showing up, or we'll never know what they look like. We'll never know what the cells look like, but we, you know, a ton of what we know about in terms of the microbial life out there, we have genomes for, but we don't have visualizations for. And I think as we start, so fungi are hard for a variety of reasons, but I mean, eventually those bottlenecks will go away, right? And so we're gonna understand how to sequence DNA even at very low levels from really recalcitrant organisms or you know, RNA. and it's just a matter of time. Whereas in terms of the fungal morphin, and I you know, hopefully I don't come off as thinking that it's completely useless. I think it's the morphological things are very useful. And in terms of classification, it's good and great. Fungi are especially, I mean, I think they make an especially good case for why you need some sort of bridge, though, because we have all of these other species that are classified differently in their asexual and sexual forms. And there's, you know, that kind of divorces the literature from itself as well. And it's, yeah, it's,
I think
very
okay, here we go.
interesting. I think we're like in the middle of this changeover, and it's not going to go pleasantly. Thing in out more taxonomy goes pleasantly. So this is like you said, where it's easy to get the DNA.
We just had a very interesting, like, Dave really slow and then Dave really fast. It kind of went like you had a lag and then you caught up really fast. So
sorry.
it was what is that? You had a saltation. You slowed down and then you had okay, sorry.
Okay, I want to come back. Fungi. We should talk about fungi more often in Microbiology Journal Club, because like they are so great historically for like ecological and functional gill context and hints you can take from where you find them, but they're also so infuriating for the reasons that the classical taxonomies for fungi are infuriating. And I agree with Dave that technology will probably redeem us in the end in terms of, you know, revising a fungal tree of life. A paper was just published on that recently and makes me very guilty about things I've said about the Mucora mycotina, sort of. But let's
wait, I'm sorry, I can't let you go by. Did you insult a group of fungus and now you feel bad about it?
No, I think I call them the wrong phylum.
Oh, that is insulting them though. So
yeah, it is. It is insulting them. But I mean, it's the Silva taxonomies. That's what I'm going to blame, and that's what I know, because I don't spend all my time culturing Umbellapsis and Mortirella, which I probably should, because they sound like they're really interesting. They probably, well, they're all interesting. So yes, I did insult a group of fungi. And I still feel bad about it, and I'm not sure how I'm going to handle it on my next paper. But let's talk about the parable of Pseudomonas syringae. I actually thought syringae's name did not come from lilacs. I thought it came from having a type 3 secretion apparatus, which is dumb. That is actually what I used to think. It had a syringe, okay? So this is what I thought about Pseudomonas syringae back in the day. Now I know differently. Okay, so
I was there once too. I had the exact same thought originally. So
I feel less alone in the world now because of that, and also because of fungal frustration. So this is really interesting, this play-by-play that Dave gives in here, where, you know, Pseudomonas kind of hangs out on plants. It kind of hangs out everywhere, really. But starting on page 434, talking about this attempt at sort of the low-pat diagnostic scheme, which was a mishmash of enzymatic characteristics, being able to make potatoes squishy. What else was in there? Can I just ask about this tobacco hypersensitivity thing? Because like I didn't look at that. So Dave, tobacco, explain.
Yeah, so if you stick if you take a syringe and inoculate most isolates of Pseudomonas syringae into tobacco, it'll, at high densities, it'll cause basically a hypersensitive response. So it's like the R genes, but we don't quite know what's doing. It's something maybe in the syringe, the type 3 secretion apparatus itself is triggering kind of a defense response, but you basically see the tissue collapse as an immune response. And so it was diagnostic because all of the pathogenic isolates syringae showed up doing that if you inoculate them at high levels. Not all isolates do that, but that became part of the scheme because when people originally cared about it, they cared about the plant pathogens, and the plant pathogens always did that. So
interesting perspective there again on how stakeholders shape where the classification scheme goes. So LOPAT happened, and then we move into pathovar. So we'll just talk about who it's infecting, and then green beans came along, and Phaseola cola was not necessarily Phaseola cola, depending on what you wanted to name it. And you've given that play-by-play of the DNA hybridization that you're trying to do with strains. So I'm assuming that you're talking about criteria all up in here for telling people when a species is a species. Is there a nod on that? Is that what you're trying to do right now?
Yeah. So it's this complicated, somewhat antiquated process, but basically, in order to truly name a new species, you need to publish in the IGA-SEM, and then in order to publish in IGA-SEM, in order to name truly new species, you need to do a bunch of different phenotypic characterizations and molecular characterizations based off of this 1990 and a couple amendments to it, Handbook of Bacterial Taxonomic Nomenclature. And so Dave Coyle has a good blog post about how that actually happens, but it's still, we still have to do things based off of the early 90s and what we were able to do then based off of a variety of different characterizations. You can, and this is where it gets kind of weird, because then you can write up new species names in other things, but they're not quite official until there's a note written in the IJSEM about, like, hey, here's a new thing that's, we're making a note of it. But other than that, it's, there's kind of one official way to do it according to the taxonomy societies, and a couple of other ways that you can do it in a less complete way. And then there's people that just do it in their own papers and publish the papers without regard for like official status. So that kind of muddles things as well.
Well, but part of the problem too is that, you've got, and I think you mentioned this, the Candidatus distinguish, because I did my postdoc on bacterial endosymbionts of insects. Like, you're not growing that. Like, you can't just pull that out of an insect gut and like plop Buchnera or Blackmania into a flask and like get a happy culture of stuff that you can then do phenotypic characterization of. So it's always a little frustrating when you're like, well, what do you do if you can't grow it, or it needs all these things, or you haven't figured out the tissue culture for your ant yet, which is not what I wanted to work on.
There was an instructive Twitter discussion maybe a month ago. So I forget how it went down, but somebody went in and reordered a phylum in bacteria, and renamed it, or kind of redesignated. So the Banfield Labs, maybe it was Archaea, one of those really deep branching clades. There's a paper that kind of renamed, I think it was the level of phylum, without actually doing it in an official way. And it's kind of anything candidatus and non-cultured and just metagenomes, it's totally like wild west in terms of taxonomy. And so there's really no official status. And so people are trying to figure out now, like, how do we actually keep account of this without worrying about losing all this information to just a muddled argument of different labs fighting over what to call things. Like, although I think that the resolution somewhere, the bright side of this is now that there's a bacteria called Hugabacter. So
yeah, there you go.
I know.
Yeah, but I mean, every time I teach symbiosis, I have to re-ask, are we still calling it Vibrio fischeri, or have people decided that Alivibrio fischeri is what we're going? But but when I ask, the Vibrio fischeri researchers are like, we're not changing. Like, okay.
Well, yeah, and Burkholderia is now Paraburkholderia. And rhizobium, the nodulating rhizobiums are all in cipher now. So it's like,
it's a mess. It's a mess.
Okay, so on that theme of messiness, let's keep going with taxonomic and phenotypic classification hell. So I have this note on page 435 that just says, Pseudomonas syringae lives in too many places and does all the things. So phenotypic classification equals a bad time. Which is pretty much summarizing that paragraph. So Sensu Latu is a model system for environmental genomics, but this is like typecasting. I mean, I know it's not the movies, but I mean that's the danger with these connotations and names, right? But we don't really know if people are just calling things Pseudomonas syringae when they go and stick things in GenBank just because it's convenient. The 16S told us that this.
Well, so you'll get it from like RPOD or the MLST. So 16S won't give you syringae per se, but usually to get it in GenBank, you're doing some of the MLSA loci as well. I mean, that'll place it pretty well, but then it becomes a question of if you get it if you find it in a cloud, like, I mean, you can't give it a pathovar because there's no pathovar for cloud, right? So then it doesn't have a pathovar, so then it's just Pseudomonas syringae. And so then you end up with a bunch of Pseudomonas syringae that doesn't have pathovars, even though that cloud bacteria is virulent on a host that you just haven't tested it against. And so what I've ended up doing, and I think what a lot of people end up defaulting to is in the absence of like definitive taxonomic ways to classify things, you just call it Pseudomonas species. Because you do, the further you get out, the further you get away from like the canonical Pseudomonas syringae type strains. And so you call them Pseudomonas species, and then that happens, but then you see other people in other environmental pseudomonads doing the same thing, and then you end up with a bunch of things called Pseudomonas species in GenBank, where there's no, and then to truly bring it back to the teaching in the classroom, you'll now see there's papers coming out from all around the world, and these smaller research groups or research groups without necessarily the capabilities to do wholesale like characterizations, but they'll find something that's fluorescent on KB media and it's from a plant, and they'll either call it Pseudomonas syringae or they'll call it Pseudomonas species, but there's no framework to place that against all the other strains. And so like it's hard to actually go into the literature and find papers from places where you wouldn't necessarily expect them that are actually talking about cool new isolates of this bacterium that I care a lot about, because there's no linkage to the name there. And if you don't know the group and you don't know to look out for them, then you don't know what to search for in terms of like what the new paper is. And so if you have a new disease, there was a new disease on citrus that was from the phylogroup 13 strains of syringae, I think. But they called it Pseudomonas species. And so you lose the fact that it's probably within that realm, but you would actually have to go and look and find the genomes and search for the genomes and then go back and find the papers from the genome entries to know that it was actually something that was worth looking at. And so it's a problem because there's a lot of information that gets lost to just people calling things different things. And never mind I probably have to go soon, but I'll just make a plug for actually Laura worked at the USDA, so she can back me up on this. Species names are hell when it comes to actually doing regulations and actually sending strains to people or trying to figure out what strains can import, especially with, there's some xanthomonads and some Ralstonia that are on the select agent list now. And so, okay, like how do you, if you find something and it's on the select agent list, like you have to report it. But, is it really Xanthomonas oryzae, or is it a close relative that by all intents and purposes looks like oryzae, but it's not really? Like it might just be something phenotypically slightly different that's related. And so, from a regulatory standpoint, it ends up being a bear because, the USDA is like 30 years behind changing names on things that even worse than we are now. So it's, and I don't see that getting any better in the near future. But.
So I want to say that this hits on some really interesting things because I mean, in my papers, I'm working with the amplicon sequencing data, and you conservatively just, because of the way I've handled data, I'm dealing with describing things at genus level, and it's a conservative approach, not unlike what you were saying, where people report Pseudomonas species. And there's defensibility in that for not wanting to overinterpret or misassign. But then from the other side, as you've just mentioned, with policymakers who need precision of identification to defend decision-making processes, it's still not sufficient because certainty of identity may not necessarily be established. Real concern for something that makes it on a list if we haven't had enough, well, maybe there is enough, maybe there's a framework for reproducibility of associating it with a pathogenic event may happen, but there's imprecision from both sides, like naming and then it not being the thing, or not naming but wanting to know if it is the thing, is a problem. And I think with that, I want to move to where genomes can kind of equalize precision in what we point to independent of a name, which is this figure here. And it's really nice because it gets into Marrakebi et al.'s genome similarity classification. So I'd already pointed Laura to the PLoS paper that you cite that actually walks you through how you do a position-based thresholding and assignment of genomes where you've got these appended numbers for genome regions. And that's basically what is going on here. I'm a little too freaked out by the time constraints and also just, again, my head's sort of melting to sort of walk through the diagram. But what we see here are some P. syringae type strains. I think you've set DC 3000 as like the reference genome. And then what you see to the right of it, which is walked through in the description of the paper, and even further walked through in Marrakebi et al. That's reference 52 in Dave's paper. How you create like sort of a subscripted ABCD positional scheme where each of those letters represents a region of genomic similarity. And then you can assign a number that actually designates, gives you sort of an overall view of regions of genomic similarity between the things on the tree. So basically you start with your reference and you would take another genome, do the walkthrough, assign your thresholds, iteratively go through, assign for the next genome in line. I think I'm getting this mostly right. And what you have is, it allows you to build from the ground up every time you need to talk about something, a comparative genome similarity based on who you've chosen as a reference. And this is actually, I think, one of the ways that people are always going to push back on things they've always done, and people are definitely going to push back when policy has to be completely ripped apart and restructured. But the nice thing about this is if you have well-characterized strains with clinical or agricultural pathological or biodiversity conservation prioritization type connotations that happen to sit as being organisms with a characterized genome, then you can take this thing that has a context and use it as a reference genome and just be like, look, we're pointing to these things that have genome identities, and then you can tack on the data on the end that says, well, we found it in a swamp, we found it in a necrotizing sore on the back of a cow after rainfall. You know, all the other data that goes with where these strains are, what they seem to be doing, can be pointed to around the strain that has now been identified at genomic level similarity to a reference. And we're already struggling with handling all the other pieces of data that dictate when a 16S data set was collected, trying to encourage best practice so that it's not just etiquette to fill out me-marks forms, but having ways to organize this. And the thing that Holly's paper gets back to, and one of Holly's research interests, which is the idea of refining classification through informatics methods and visual systems for providing this composites of data that has different appeals and different levels of importance in a multidisciplinary context, or a policy context, or a research context, or an educational context, can be unified along sort of an objective axis that's defined by genome similarities. I think I'm just going to open the floor at this point to comments about that summary so that I can be corrected if I got things wrong, or that people can sort of bring in their reflections on that statement.
Well, so just to think in terms of a unifying database, when Dave was talking about, well, it would be really nice to be able to classify certain features where you could just choose whether or not it was positive for a certain type of test or had a certain phenotypic feature, and you would be able to search that in a database. I think that would be a great idea in terms of like if you had a giant collection of all of it together, like if you're not able to bring the collaboration of the scientists together in terms of studying these specific things individually, if we just had a gigantic database where everybody put in what they found, phenotypically it'd be like, okay, for this trait it's this, for this trait it's this, for this trait it's this, and then any researcher could just go on that database and just search the specific phenotype that they're looking for. And then you don't really have to have a name, because then you would have the name that it's been given, whether it would just be Pseudomonas species, but then you would have all of these things that you would be looking for, and you can just choose that out and then probably go contact that person and be like, hey, I think that you have something that's of interest to me because it has all of these traits. And so if we're not like in an age where we're able to collaborate in terms of having large databases and information on the internet, I think that should be an advantage that we can use this for instead. And then that in itself will produce more collaborations, because you'll start to build connections between people when they know that, okay, you probably have this strain, it doesn't have a name, but it has these characteristics. So can I get it from you? Kind of like in that sense.
Yeah, Karen, I think that's a good point. And I think Nicole, you've made some. I liked your summary. It was a nice kind of broad summary of your goals and what we're trying to do moving forward. And I think both articles made the point that, you know, this is something for Holly's piece. Her urgency is about the fact that taxonomic experience for non-microbes, you know, we're talking now about like nematodes and insects and jellyfish and platyhelminths and stuff like that. Our taxonomic expertise, the people who've spent time working on that in specific, it's starting to dwindle. So people are starting to retire. We don't have as many people coming into those fields. So the urgency with Holly's piece is to make sure that we don't lose continuity with what has been established, what is a major body of knowledge that we want to continue to be able to access. And I think the urgency with Dave's piece is that we are drowning in genome data, and we're drowning in strains, and we need to figure out how to organize all of this information so it doesn't continue to be, like somebody mentioned, a morass of, you know, like, so it doesn't continue to be just this quicksand that prevents people from actually capitalizing on all of the sequencing that we've been doing, all this data that we're generating. That's my thought.
I think I just wanted to
say, part for me too is just to imagine going forward that it's only going to get worse. And so it's great to have these traditional schemes where we have experts that get to designate what gets called what, and there's a gatekeeper to aspect to it. But we're going to have a lot of people out there just sequencing and doing other things and isolating new things, and there's not going to be the experts regardless of how many people we train. Like, we're just not there's not going to be an expert for every taxa like there once was. And so we need to develop ways to be able to classify. And I like the Lynn's framework, because it is that the key is that it's relational and expandable. So if it's relational, you don't have to rewrite everything after that first time. You can designate whatever the reference is and then base everything on that reference and then call it some characterization. And it expands however much you need it to expand. So you don't actually it'll expand regardless of how much data shows up. Versus like the species naming now, it doesn't do that, because you have to sit there and actually, ID everything and classify everything. And so that's my daughter.
She's got thoughts too.
All right, so I'm gonna go.
Bye, Dave. Thanks for joining us.
I have to run too. It was really nice listening to your discussion. Thank you.
Thanks, Luke. Okay, I'm just going to say one thing that sort of is a last thought about sort of the classical taxonomy business with this idea of the deluge of genomic data, and something to sort of bring it back to education and this idea of expertise, which is that in every 100 level microbiology lab, there's, well, and maybe 200 and 300, there's that aspect of culturing and identifying the unknowns. And a lot of students get it wrong, which I mean is a very clear teachable moment about what that expertise looks at, because they're just kind of plunked into it. So that is one of those opportunities to get people to understand how hard it is to actually cultivate that expertise in the microbial sense. The second part of that is coming back to the informatics. So Karen, your idea of the database combined with, I think, some ideas. So I think there was that paper, PaperBlast, which is like a search engine where I think you can put in protein names and it pulls all the references. So using text mining to actually sort of link, maybe wrong, maybe right, designated organismal names with the literature that's out there, so that researchers, based on their knowledge, content knowledge, context knowledge, physiological knowledge, and ecological knowledge of where they're seeing things in their system, can start to think about where do we look for more genomic information if we need it, where do we need more physiological information if we need it, as well as imaging technologies. And this is why I kind of wish Kendra was here, because she's participated in a couple of BioBlitzes, and they have all those like apps on phones where you can take a picture of a mushroom and there's a database. So there's also software that has dichotomous keys built in, but also systematic ways of doing image capture so that we at least have a digital archive, you know, that has an insect at a certain angle so you can see symmetries, or you can see plates a certain size, so that data can be kept until somebody can do a partial job of taxonomy, or until we can use size recognition maybe somewhere down the line, or 3D printing to actually get a feel for how does this match with a historical record of where genital plates should be on an insect. So looking at ways that we can leverage technology in a big way to do automated decision making or semi-automated expert assisted decision making, to keep some of these classical taxonomy approaches feasible and continuous, alongside training the body of people that our academic systems and our conservation systems and our government systems will be able to sort of subsidize and train. So I think, and I mean, especially with the genomics data, we already know that there's a high technology interdependence and a data management literacy that has to be respected. And I think with classical taxonomy as well, there is an order to collecting the data and managing it. And I think these hygienic aspects, they're not very romantic, but they're also a key piece to bringing all of this together, as well as thinking about things that Dave has endorsed, which are systematic genome-based classification. So I will wrap up what I have to say there, and Laura, I will hand it over to you.
Okay. Thanks, Nicole. That was great. I think you did a really nice job leading this. And it was a broad and kind of, a rather freewheeling topic. So I think that was well done. Karen, if you don't have any final thoughts.
I was, do I remember what I was? Okay. I was just going to say that in terms of like, all of these things require massive community collaboration. And either like on a scientist, like a grassroots, from a grassroots movement type of point of view, or from a government instituted type of view. So it's either, it's got to come from top down, or it's got to come from bottom up. And I think either way, that's really important, that in order for these things to get implemented, they need large amounts of funding, large amounts of support. So it's good that both Holly and Dave are writing these types of papers, because as the message gets out there, then people will be like, okay, here now, what do we do about it? And start to move those things forward.
Yeah, I think that's a great point. I think that part of the purpose of these perspective pieces, that was a lot of alliteration, is to try to kind of foster a groundswell of support for this, so we're not just kind of stumbling down the road. We actually have some direction in where we're trying to go in the future, so that hopefully somebody takes up this challenge. It will not be me, but I will gladly march alongside if somebody comes up with something. Okay, so with that, we'll close. Thanks to everybody who joined us, and we do this every month. So we'll be looking for papers for next month. If you've got a paper involving microbiology of any kind on your, we'll talk about fungi. We will, I promise. If you've got a fungi paper, we'll talk about it. But if you have a paper on your to-read list and you'd like to suggest it, please feel free to send it to me. I'm at microwaves sci, so like microwave plus an S plus sci. I don't know. And we can put it on our list, and we'll be back in late October for our next Micro JC. So with that, we will sign off.